So good afternoon, everyone. So I'm Christine Kubiak, the operation director at PedCRIN, and it's my great pleasure to welcome you to this third PedCRIN webinar, which is related to the introduction to neonatal tubes. I will give a very short introduction of about the PedCRIN project before giving the floor to our moderator and speakers. Just a few information about the PedCRIN project. PedCRIN stands for Paediatric Clinical Research Infrastructure Network, and is a project funded by the European Commission, so Horizon 2020, that started in January 2017 and coordinated by PedCRIN. This project involves 15 partners from 13 countries, and you can see on the right-hand side the list of the partner. The objective of the project is to enhance the capacity for the management of multinational pediatric pediatric and neonatal non-commercial clinical trials. And during the course of the project, 3 clinical trials were supported and conducted. The project consists of 6 work packages, and I just want to highlight 2 of those work packages. The one related to the development of the tools for the setup and management of neonatal and pediatric clinical trials, and we will have an example of the achievement during today's webinar, and also the communication working group, including patient and families empowerment. Just for your information, so the project is ending on the 30th of June, and the final event to present all the achievements during the project will be held on the 16th of June, and you can find more information on the PedCRIN website. So now, without spending more time on this short introduction, it is my great pleasure to give the floor to Donato Bonifazi, who is the Chief Executive Officer at CVBF and one of the work package leader in the PedCRIN project. And he will chair the session and also introduce our speakers today. Many thanks to everyone, and enjoy the webinar. Thank you.
Thanks a lot, Christine. I'm very happy to introduce today's speakers. And first of all, I would like to thank you and all at PedCRIN for this very good organization of this series of webinars, hoping that we will have the chance to have even more webinars together in the future, because PedCRIN was a very good example of collaboration of an existing research infrastructure as PedCRIN with a dedicated community to, in this case, pediatric clinical trials, coming from, as you explained, all over Europe with 15 partners. So I'm representing the partner from Italy, and I was leading the work package 3 together with Evelyn Jacques-Gren for the development of pediatric and neonatal tools. We already had a webinar on the pediatric tools. Today we will listen to the presentation of the neonatal tools developed within the PedCRIN project. So it's a pleasure to give the floor to Evelyn Jacques-Gren, who will start the presentation and will better than me explain INSERM role in the project and the very good tools that were developed. Thanks a lot. Please, Evelyn.
Thank you very much. So what I'm going to present now are the tools in neonates. This was the objective of the work package 3, to develop tools specifically to neonatal trials today, focusing on neonatal specification and based on the identification of the need of the scientific community. We elaborated these tools and we are now in the phase of disseminating them. They were grouped in two different parts, one related to the procedures for the setup of neonatal trials, and the second, procedures for the management of neonatal trials. Next one, please. There are a strong rationale to the development of these tools. First, the neonatal population is heterogeneous in terms of age. First, gestational age at birth, and we have post-term, term, and preterm babies, already divided in additional extreme preterms to late preterms babies. There is also an age to consider, which is postnatal age, and the combination of two, which is the post-conceptional age. But in addition to age, there is a high variability in weight, and we need to identify small for gestational age or intrauterine growth retardation with a major impact on maturation. Here is, for example, the example of a population PK studies that we conducted, which illustrate how variable the babies can be, with weight ranging from 400 grams to 11 kilograms at birth, and gestational age, for example, from 22 weeks to 42 weeks, which underlines again the major heterogeneity of this population. Next. In addition, it's a high-risk population with at least 50% of all neonatal deaths related to preterm, either directly or indirectly, with complication during the intrapartum period or with, for example, neonatal infections. Next. In addition, neonates have unique disease and risk factors, unique disease such as respiratory distress syndrome or patent ductus arteriosus, and unique susceptibility responsible of the high risk of death related to necrotizing enterocolitis or retinopathy or intravascular hemorrhage, etc. Next. The rapid developmental changes that occur with growth and maturation have a major impact on the pharmacokinetics and pharmacodynamics of this drug. And this has an important impact on dosage and effect of these drugs. And this has to be also considered. Next one. There are many additional challenges to improve therapy. We need to conduct PK, efficacy, and safety study to obtain informative data. We need to have validated analytical technique for drug measurement. We need to use adapted methodology like population models, simulation extrapolation, and optimal trial design, respect ethics and regulatory requirements. And at the end of that, neonatal drug trials are really required and urgently needed because marketing authorization in neonates are limited. From 15 to 60% of drug used in neonates are off-label. And in the absence of dose evaluation and validation, there is an increased risk of underdosing or overdosing, an increased risk of lack of efficacy or dose-related toxicity. In the absence of adapted formulation, there is also an increased risk of medication error. And as illustrated here, the doses that are given to neonates are highly variable. This is, for example, shown here, the example of amikacin or ceftazidime, showing that the minimal or maximal dose, daily dose that are given are highly variable between centers. but in addition, do not really follow the recommendations that are provided in NeoFACS or the Red Book. And in the next slide is illustrated how variable the doses of cefotaxime are given in the French centers, from 180 milligram per kilogram per day in 3 divided doses up to 50 milligram per kilogram per day in 2 divided doses, showing that the data available to use and validated to use this drug are not available for neonates. And last but not the least, neonatal drug trials are really lacking. And in these studies that we published a few years ago, we observed that only 0.2% of the trials that are registered in clinicaltrial.gov are related to neonates, although there you can see here quite an increase starting in 2004 and related to the implementation of the different pediatric regulations. Next slide. And this is confirmed by the EMA 10 years report, 10 years after the pediatric regulation, showing that the number of PIP up to more than 700 is quite high, but that those including neonates are only 25% or a bit more, and 67 were on PedCo request, which shows that there's still some way to go before obtaining data for neonates. Next slides. So in this context, we developed neonatal tool using the following methodology. We first contacted experts in neonates, experts in trials, and we performed a literature review combined with a gap analysis and combined also with the experience and mission of the PedCRIN partners. And the tools were developed, as I told you, in 2 different groups, tools for the setup of neonatal trials and tools for the management of neonatal trials. In the next slide, you see how important it is to have a multidisciplinary expertise to develop this tool, to answer regulation requirements, to use innovative methodology, to manage pains, to handle biosamples, and all these issues are listed here. And in the next slide, as I previously told you, they were grouped in 2 different sets of tools. In procedures for the setup of neonatal trials, we identified 5 topics: methodological aspect, pharmacokinetics, patient-centered outcome measures, design aspect, and ethics during the setup of neonatal trials. And we worked on 25 tools, and I just presented 2 of these tools in the next slides. One, this tool for defining the target population, there were 3 tools, 3 tools: standard age group, sample and data collection, and analysis. And for that, for the neonatal age, as we know that this is a proxy for maturation and covariate influencing PK, PD, and efficacy, we used the reference documents and standardized definitions that are presented in WHO document and the FIGO documents. Next slides. The definition of the different neonatal age and age group is really important. And in our experience in neonatal trials, it's quite frequently mentioned age without any precision of what age is required, which is very, very confusing and difficult to interpret data. We also develop an important guideline for blood sampling during clinical trial research in neonates. and listed some of the very practical issues related to sampling. I'm not going to discuss all them, but this is a practical presentation of the documents that were released, for example, by EMA and published in the literature also. It's important to follow all these practical issues to be sure that the practical issue in sampling are correctly followed. Next slide. For the setup of clinical trials, there is also an important issue with pharmacokinetic studies, because we know that gestational age and maturation has a major impact on drug disposition. So in the next slide, we developed here 4 tools: study procedures for pharmacokinetics, innovative population pharmacokinetics and use of scavenged samples, population pharmacokinetics and extrapolation, and point to consider in neonatal pharmacokinetic studies. We also identified 6 topics with 25 tools for neonatal pharmacovigilance, biosamples in neonate monitoring, neonatal clinical trial conduct according to GCPs, training in neonatal trials, and managing clinical trials. And these are all groups in procedures for managing of neonatal trials. Next slide. I just want to finish this presentation by talking about deliverables 3.4, which aim to define the criteria for certification of neonatal and pediatric clinical trial units. That is a work that we conducted in collaboration between INSERM, AP, and CVBF. This work, in the next slide, is very important, as clinical trial units has to support investigators in providing statistical, epidemiological, logistical, and methodological expertise and help in coordinating trials. And this, as you know, is very important in multi-center trial and multinational trials. And there are many activities that these CTUs have to have in charge related to planning, conduct, and reporting. And without going into detail into that, next slide, we discussed and considered 5 different areas. To develop this tool and this first document, we first considered the specificity of research in children, the need for experts that are required to support the trial. We also analyzed the literature, and we took advantage of the experience of PedCRIN in establishing the criteria for data center certification. Next slide. And with this, we provide an overview of the neonatal pediatric expertise that CTU may require in order to support neonatal trials. These criteria might be used to develop or to organize a neonatal and pediatric CTU network to support these clinical investigation centers and research centers. Although we know that local organization may vary between CTUs, and that, of course, all neonatal pediatric CTU that need to provide services cannot cover all these services, but may rather focus on selected activities. Next slide. And the documents that was sent to the Commission under deliverable 6.4 is a first version of a proposal that has to be evaluated, that has to be expertised, to be able to respond to all the requirements of these CTUs. It will be then developed, and we will work again on this to have a finalized version. Next slide. So in conclusion, Neonatal research requires involvement of many, many experts, and we try to put them together to develop these tools, and we know that they will have to be commented and maybe improved. They cover 11 topics. Not all of them are finalized. These 11 topics including 50 tools. And as you know, and I just tell you, CTU criteria for neonatal trials are under finalization. Thank you. Oh, as a conclusion, I listed here the key points to consider when we need to put and organize trials in neonates. First, analyze the scientific aims, perform regulatory evaluation, consider ethics, feasibility, contact research units with expertise in neonates and in children, perform recruitment, contact parents, and have a very strong sponsorship. Thank you for your attention.
Thanks a lot, Evelyn. So your great experience in clinical research is clear that was addressing very great results with these tools. And now Beat, which joined your, let's say, research unit 5 or 6 years ago, but she's experienced as well, and she will present herself and detail several tools that were offered within the project. So Beat, thanks a lot for participating to the webinar, and the floor is yours.
So I am working with Professor Jacques Séquin for some years now, and I'm very happy to present here. By training, I'm a pediatrician and a drug safety physician. So this session, I will take advantage of having the word, so to speak, and we'll talk mainly about drug safety within these tools. I've selected 3 tools, which I think could be interesting for you, because talking about drug safety and how to manage pharmacovigilance and risk management in neonatal trials can be challenging. I suggest that I have a short break after each presentation to see whether there are any questions that need to be addressed directly. If not, I will continue. Okay. So the first topic is drug safety and protocol development in neonates, and I've just provide some brief points to consider. So when we talk about drug safety in clinical trials, evidently they are a part of the objectives. They're usually secondary objectives. And you can divide them into those that relate to the different protocol sections and other trial-related activities. And here I have just listed a few protocol sections which may be touched by drug safety questions, which may not always be evident. So for example, if we talk about the benefit risk of a treatment, of a new treatment or any treatment in a protocol in the introduction, evidently the word risk means side effects. In the objective, as I mentioned, it's usually a secondary objective. Exclusion criteria are very important, and there are a few others listed here. For the trial-related activities, ethics boards and regulators are very keen to understand the risks involved. And obviously the regulatory obligation to report adverse events or adverse drug reactions to the relevant authority is important. So when we think about safety, it doesn't come from nowhere. It sometimes seems like that, but it isn't. It's actually evidence-based, and it's comes from what we call the safety specification. So when we're talking about neonatal trials, it's the neonatal safety specification, and it is evidence-based risk management and pharmacovigilance in clinical trials. So with the safety specifications, once you have that, you can build much easier plans for risk management and pharmacovigilance. The safety specification combines safety data, non-clinical and clinical. It can come from many different types of populations, and it combines this information with the understanding of the neonatal study population. The safety specification is a living document, which means it changes over time. So as more people get exposed to a given treatment, you have a chance of finding new adverse drug reactions. In safety, we identified and potential treatment-related risks. So identified is something where we have solid evidence that this is a side effect or adverse drug reaction. And for potential ones, where there is some evidence, maybe some preclinical or from other populations, but we still need to confirm that this is actually a treatment risk for the population in question. And then importantly, we also talk about missing safety information. So for neonates, it's similar to the lack of pharmacokinetic data. There is very little data on treatment risks in neonates. And as Professor Jacques-Legrand mentioned, understanding pharmacokinetics and pharmacodynamics in neonates is important, in particular for dose-related toxicities. So here you can see on this slide a brief graphic of how this all fits together. So on the left-hand side, you have the so-called neonatal safety specification with the identified and potential risks. Added to that, because of the population, we also have excipients and medication errors, and also safety information that is missing. On the very right-hand side, you have the characteristics of the neonatal study population. So for example, what are the specifics of adverse drug reactions in neonates? It's not the same as in adults or in older children, evidently. And the awareness and understanding of reference values for the population, which also do change. They are not necessarily or almost never the same compared to older children and adults, and they change in different age groups and risk groups in neonates. So this is very important to take into consideration when we think about safety. So you put the left-hand side, the neonatal safety specification and the characteristics of the neonatal study population on the right together, and these two arms of information or leaflets of information inform your neonatal trial protocol. So you can divide the neonatal trial protocol into risk management and pharmacovigilance. Risk management is a proactive activity with the idea of reducing the risk of side effects and their severity and poor outcomes. So it includes exclusion criteria, dose reduction and holding criteria, discontinuation instructions, treatments that should not be given, and obviously the monitoring of patients for safety's sake. And then you have, on the other hand, pharmacovigilance activities, which can be general or are general. So we look at all adverse event data or lab data, anything we can get our hands on, and risk-specific safety data. So we look, for example, if you have a risk of, say, abnormal liver functions, then we may monitor liver function, for example. So we have risk management and pharmacovigilance all in one in neonatal protocols. So from a practical point of view, how would one go about when we think about writing a protocol and safety. And one way of looking at it is summarized in this table, and I encourage you to go into the tool to read more about it. But here you can see on the left-hand side that there are many different protocol sections, including the design of the case report form, which are touched by the understanding of the safety profile of a drug. And it is important to anticipate these things as much as possible, because if this is considered after the fact, it's more difficult to understand what actually happened. So just to summarize on the issue of drug safety and neonatal protocol development, it is really helpful to have a neonatal safety specification, which basically summarizes all the relevant data. It helps with proactive risk management, high quality safety data collection, and data-driven benefit risk assessment. It is useful to remember that neonatal trials are in and of itself rare. So having the opportunity to collect high quality data is a unique chance which should not be given away easily. So when a team is developing a neonatal protocol, it is useful to have an experienced pharmacovigilance physician easily available. They can help with anticipating some of the risks and manage them accordingly. Next one, data collection and analysis in neonatal trials for safety. Not easy, as you can imagine. So again, it's based on the safety specification for neonates. So this is the building block. There you will have identified what are the adverse reactions that can be expected in this population, and those there where you may have some questions, and also those where you may want to have more understanding, for example, risk factors or outcome for safety issues. And as you can see on this slide, the data collection and analysis are population specific, and as you've seen in the photos of Madame Egan's presentation, you can easily understand why they need to be population specific. Not even neonates, but depending on the, for example, the gestational age or the weight or both. So it is very specific. And it is also important to have these subgroups in order to understand what are, say, risk factors. For example, prematurity, extreme prematurity is a risk factor for adverse drug reactions, certain adverse drug reactions, or confounders. For example, if a baby dies in neonatal trials, it is important to understand that death is unfortunately common in neonates. The sample size is an important issue for neonatal trials, and changing reference values. The excipients are another challenge where we have actually a separate tool on this. Medication errors are quite common, specifically also for co-medication. So neonates who are in intensive care, they require many different drug treatments, and many of them are not made for neonates, so the doses need to be diluted, and the medication need to be prepared for the neonates. And you can imagine that in a very high intensity work environment, there is a risk of medication errors. So the next slide talks about the safety specification in neonates. As I mentioned before, it is evidence-based. It helps with risk management and pharmacovigilance, and it combines the safety data of with the characteristics of the study population. It's a living document, and it is important to understand PK/PD. So PK/PD in neonates is almost married to safety. So it's really important to have an understanding, and the lack of pharmacokinetic, pharmacodynamic data in neonates makes the assessment of safety in neonates challenging. Sorry, next slide is, so when we talk about safety data collection in neonatal trials, as in all other trials, there's a continuous safety data collection. So if at any point in time an adverse event occurs, it is assessed, it is reported or not to the regulators immediately, depending on the seriousness, and at predefined times. So for example, at study visits, or say at certain time points of the study. So we have both continuous and predefined times for data collection of safety data. It includes our understanding of identified and potential treatment risks, missing safety data if we can, then any new suspected adverse drug reactions. So if you have, say, a rash coming up and this hasn't been reported before, then this could eventually become a new adverse drug reaction observed. It is important to be clear about potential confounders and risk factors for adverse drug reactions. Collecting data on co-medications, as mentioned before, because of excipients and medication errors is important. And the idea of data collection in neonatal trials for safety's sake is to protect study subjects. So if you see an adverse drug reaction or something that could be an adverse drug reaction, obviously you will consider changing the dose, stopping, treating, whatever. And also to collect data on treatment-related risks. So there are two objectives: protect study subjects and collect data. And then the data, once the data is available, it will be included in the analysis of the safety data. And for that, you will have the safety analysis plan, which is often part of the protocol. So when we think about safety data analysis plans in neonates, obviously, as mentioned before, it is based on the identified and potential treatment risks, any data that has been collected on missing safety information, any new ADRs, and also what we include in safety is what we call designated medical events. There's actually a list on the EMA's website, which is regularly updated, and it is a list of events or things that may occur in any given population, which are commonly or often associated with adverse drug reactions. For example, Stevens-Johnson syndrome or something similar events. When we look at safety data, we look at all adverse events, regardless of severity, seriousness, duration, causality. We include everything. And then we also look at the data of test results, investigations, say blood pressure, electrocardiogram, blood tests. So we look at all data, and you can easily imagine if somebody develops low platelets or thrombocytopenia, this may not show immediately in a clinical picture. So it might be only detected in blood tests. And so if you look at a population where there is a risk of thrombocytopenia, you will have more subjects exposed to the drug that may cause this event when you look at the blood tests with low platelets compared to the clinical signs of thrombocytopenia like bruising or bleeding. And once we have done the safety analysis, we then include this information into the existing safety profile. So it's almost like a cyclical event movement. So you analyze the data of the clinical trial, and then you consider if the existing safety specification needs updating. So on this slide, I have just briefly summarized some of the challenges we may have in neonates with regards to reference values. Reference values may, as the child adapts to the life outside the womb of the mother, may change. So here I have listed just some values which could change: hematology, biochemistry, and heart rate. And this is important to consider when we start developing a protocol, a CRF, and an analysis plan. Okay. Next slide. Sorry. And so just to summarize, safety data collection and analysis in neonates is based, as you now know, on the safety specification in neonates. So it's population specific.